Hello everyone, my name is Junren Wang, or you can call me Bruce. I'm from National Taiwan University, Department of Life Science in Taiwan. Today I'm going to show you and share to you what we found in Taiwan about the flower color diversity, which reveal there's some selective filtering due to the PB preference. Let me introduce you first. What is flower color diversity usually reflected to? Flower color diversity usually reflect to different pollinator selection because different pollinator like different kind of color, like bird, like red colors, bee with all different kind of color, fly mainly bright yellow or green colors. So these are different studies all show to you. So the flower color diversity in a local area, local region, that usually reflect the plant-pollinator interaction result. So this why they are so different in color. Must be different pollinator selected result. And also it revealed the evolutionary history because a local flora may have their species come from all different neighborhood, continent or islands. So they have different evolutionary origin. So that contribute the whole diversity of all different kind of flower colors. But previous study mainly work on continental flora. Last study is working on island data, but there are still few. My colleague and other Australia people, they work on these islands. They can find out their color compared to previously you see the continental island data. The flower color diversity, FCD, is slightly lower, perhaps due to in these islands there are fewer pollinators, girl species or different kind of pollinator. They lack different kind of pollinator and the species richness. So therefore their color mainly reflect white and yellow color. Okay, so let's focus on what I'm provided here. I'm come from Taiwan. This is the Taiwan island, which located between China, Japan, and Philippines. So it's a tropical and subtropical island, and there's a very much species richness and with high endemism. This is compared to previous study, very unique local flora. And also we find bee are the major pollinator in pollinating Taiwan species, although fly and other species also, but bee appear to be the major pollinator. And in addition, Taiwan has a very high mountain, 3,000 meter, 100 peak over 3,000 meters. So they have potentially changing their pollinator distribution along the elevational gradient. So I want to raise this important question and find out the answer why whether the flower color diversity in Taiwan correlate with this feature and into its unique patterns. Okay, let's look at the Taiwan topography. You can see if we do a dissection of Taiwan from north to south, you can see almost all mountain everywhere. So in the mountain top, this cool temperate vegetation zone is quite cool. Even in summertime, it's 10 degrees or less. In lower part of the Taiwan is almost tropical, very hot, 37 Celsius degree. So it creates many habitat and niche. So perhaps species adapt to different altitude, evolve into different colors with different pollinators. Also, it have a very much richness of the species level because just I mentioned, during the glaciation time, this light blue color are the land bridge which can connect Japan, China to Philippines with species migrate from each other. So that create Taiwan very high species richness, and also due to the high mountain, you know, during the glaciation maxima, many species, because mountaintop is too cold, so they migrate to lower area, lower elevational area. But during the warmer internal glacial maxima, this temperature become hotter, the species migrate back to mountains, so that create different species. So that reveal in the Taiwan, we have 26%, 1/4 of species are native to Taiwan. So only in Taiwan, high endemism. So this create what we want to find out, why there's so many flower color in Taiwan, whether we can find any suggestion for why so many color evolve. We sample 700 species, 27 species, much more than previous studies, and we want to find out whether the color signal play a major role in attracting the insect, particularly bee. Okay, so the bee, we should not use human color to determine flower color. So we measure the petal color reflectance from UV to all visible light, and that match to the bee vision neuron. And when doing this can be converted into the bee hexagon. That means we map all species flower color onto the bee hexagon. So you can see the distribution of flower color now in bee's eye rather than in human's eye. Okay, so first we want to test whether, just like our hypothesis saying, most of the species look like pollinated by bee, whether this color match to bee favorable neuron position. You can see the hexagon sector around 30 to 90 is the strongest innate color preference. So you can see from Taiwan, low, middle to high altitude flower color all match to the bee preference area, which is similar to other regions' previous paper. So it looked like our species in Taiwan preference by bee visiting in colors view. Okay. The second thing is we want to see the Taiwan island, whether the color diversity is comparable to other regions or not. You can see the NCP calculation is basically we can draw an outline to include all these loci and see how large this area is. If the the area is comparatively to another place, then we can say the color diversity is almost similar to other place. Okay, so we can find out the MCP value in Taiwan is comparable to Japan and Australia, although Nepal has a very large MCP, maybe due to its Himalaya origin, a very unique vegetation type. But generally, Taiwan don't have very small FCDs, just like oceanic island. So more or less similar to continental area or continental island, we say, like Japan as well. And also they show two strands, one is white arm and one is yellow arm. This is human white color in bee's eye, this green area, and human yellow color in bee's eyes in this UV green area. You can see these two arms very clear. Taiwan and Japan, they share, maybe these continental islands, they share a similar pattern when they evolve compared to continental data. Their color have some trend. Next, we want to test whether the flower color diversity have changed along the elevational gradient from high altitude to low altitude. Okay. The data show up. You can see high altitude flora have a we now convert the flower color diversity into we call flower color assembly. That means whether these colors are similar to each other. If they are very similar to each other, we can show they are cluster. If they are not very similar to each other, they show over-dispersion. So we found out particularly the high altitude of Taiwan. This is high altitude Taiwan's flower color, species flower color. they have the significant level indicate the color assembly of high altitude species tend to over dispersion. That means the species in high altitude, the flower color are quite different to each other. So we want to ask why they are so different to each other. If we want to answer this question, we need to look at their phylogeny first. You can see from the phylogeny, from these 727 species, usually you can find, although closely related species have a similar color, but they change their color very quickly, right? This is phylogeny tree branch, right? And the terminal is labeled by the color of the flower species. So they change the color very regularly, right? So this look to me, the flower color in Taiwan, they are quite diverse, but also related species are even more, even they try to diverse between closely related species. So to test this, we add the phylogenetic signal concept into this. That means we relate the flower color with their phylogeny to see whether there's a two hypothesis. Is if they have strong phylogenetic signal, that means similar related species in sympatric area have a similar color to each other. So color are clustered, but phylogeny also related to cluster color. So that means you see related species have a similar color. That means strong phylogenetic signal. Or it could be random. Okay, that means the color is randomly not related to phylogeny at all. Or the other thing is weak phylogenetic color signal. That means related species have a over-dispersed color. Okay, so if related species have over-dispersed color, that means they probably want to reduce competition for pollinator. They want to coexist in the same place, so they diverge their color a little bit in this way. So when we do this, we particularly find the high altitude species under the strong selection into weak phylogenetic signal. That means close related species tend to diverge in their flower colors by this MPD or NMTD, these two measurements. Let me give you the example like this. So these are viola species, and these are gentiana species in high altitude region. They all show weak phylogenetic signal. So they are so closely related, but their colors are so diverse. So that must be a selection force want to over-disperse their color. So the conclusion is here: flower color diversity at a similar level to other area, and also match B preference. And we also show flower color have a weak phylogenetic signal at high altitude, and that could be due to the selective filtering, because bee want to visit them, and each species sympatry in there, they want to diverse their color as much as possible to be able to let bee have a constancy on them. Please let me know if you have any questions. Thank you.